Hello students, today we are going to discuss a very important topic in biomolecule, that is nucleic acids. Already you know that the new species has some similar properties with his ancestors. If you see the characteristic, the biological properties of the new species produced has some similar character with his ancestors, old generation. So this new character, transfer of character from one generation to another generation, that is called heredity. And this hereditary character is transferred in the form of DNA. We can say it is called, that chromosome. The cell is consists of chromosome, and the chromosome may be responsible for hereditary character. And this chromosome consists of proteins and some important responsible biomolecule, that is nucleic acid. This nucleic acid responsible for the transfer of hereditary character from one generation to another. So nucleic acid generally two types. Generally it is two types, that is DNA and RNA. Mainly two types. DNA in the form of DNA. DNA and RNA. These are the two types of nucleic acid: deoxynucleic acid, ribonucleic acid. Deoxynucleic acid or ribonucleic acid. There are two types of nucleic acids. And nucleic acids, actually, if you see the composition, it consists of nucleic acid mainly consists of sugar and phosphoric acid, and your nitrogenous base. Mainly these three substance responsible formation of your nucleic acid, either DNA or RNA. Generally, two types of sugar. If you discuss regarding the structure of nucleic acid, you will see mainly it has two types of sugar. Sugar is your ribose sugar another one is deoxyribose sugar. Generally two types, ribose sugar and deoxyribose sugar. What are the structure? Generally two types of sugar in nucleic acid. One is ribose sugar, this is called deoxyribose sugar. Ribose sugar, if you see, it is that 5-membered ring having 6 carbon, 1, 2, 3, 4, 1, 2, 3, 4, 5. 5-membered carbon, and here the ring is 5-membered ring, 1, 2, 3, 4, 5. 5-membered ring having 5 carbon atom. Here also 5 carbon. Only difference is at carbon number 2 that in deoxyribose oxygen is removed. That's why it is called deoxyribose. This is called ribose sugar containing 5 carbon, and this is deoxyribose at carbon number 2, there is no oxygen. This is called deoxyribose. Two types of sugar, ribose and deoxyribose. And mainly ribose means available in RNA, and deoxy in DNA. This is the main difference between the DNA RNA. There is two types of nucleic acid, but difference in the sugar content. Okay. Then come to that is phosphoric acid. That is come to nitrogenous base. Nitrogenous base mainly two types. One is that is purine. Second one is pyrimidine. Purines and pyrimidines. And here purines mainly contain adenine, A and T. A means adenine, T means thymine. And pyrimidine contain cytosine, uracil and guanine. Adenine and guanine, AG, and your thymine, T. This is purine contains adenine and guanine, and pyrimidines contain what? Cytosine, uracil and thymine. Only the difference in the structure of purines and pyrimidines, that is cyclic structure. So two types of nitrogen base, purines and pyrimidines. Adenine, guanine, cytosine, uracil and thymine. If you write the name, that A stand for adenine and G stand for guanine, and here C stand for cytosine and U stand for uracil, and your T stand for thymine. Thymine. So this is the types of base. Okay, cytosine, uracil and thymine. So this is the thymine. This is three types, like two types of base, purines and pyrimidines. Two types of, that is purine, three types, that is pyrimidines. In short form, that is AG and COT. AG, that is adenine, guanine, and COT, cytosine, uracil and thymine. So these are two types of base available in DNA and RNA. Mainly, if you see DNA contain the base adenine and guanine and thymine and cytosine. AGTC, adenine, guanine, thymine and cytosine. But RNA contain adenine, guanine, in place of thymine, uracil. This is the difference. DNA contains the base adenine, guanine, thymine, cytosine, and RNA contains adenine, guanine, uracil. Only difference is thymine replaced by uracil in RNA. This is the difference between the base difference in RNA, DNA, RNA, and your already sugar difference. Already I have told you. DNA contain ribose and DNA contain deoxyribose, RNA contain ribose. So this is the difference between sugar content and base content in DNA. So this is about nitrogen base. Third component is your phosphoric acid. Phosphoric acid mainly contain, this is the structure of your phosphoric acid. Then another wedge. This is the structure of phosphoric acid. It is available in the DNA in the form of, it is attached to. We will discuss that bond between the phosphoric acid, sugar and base. Mainly nucleic acid, if you see the definition of nucleic acid, it is nucleic acid is a polypeptide of nucleic acid. It is a polypeptide. If you see, it is a polynucleotides. That is, you can write polynucleotide. Nucleic acid is a polynucleotide. Is a polynucleotide. Nucleic acid is a polynucleotide. Short definition. It is the polynucleotide is called what? Nucleic acid. Means large number of nucleotide groups attached to each other forming nucleic acid. Then what are nucleotides? If you see, nucleotide mainly contains, that is your sugar, then sugar plus phosphate group plus base, nitrogenous base. These three composition results nucleotide. Nucleotide is the combination of sugar, phosphate and base, all three forming nucleotide. And sugar, nucleoside is the combination of sugar and base. Sugar and base is called nucleoside. And when phosphate will added, that is called nucleotide. So what are the arrangements? Let us discuss. Here what happens? If you see the structure, generally sugar, you see sugar attached to base. Here are carbon C1. That means sugar has, already we discussed, sugar has C1, C2, C3. C1 position attached to sugar. For example, if you see ribose, if you take the example of ribose, that this one, this is the 1, 2, 3, 4, then CH2H, then 5, and this position attached to base. Base attached to sugar at C1 position, and this attachment at carbon number C1, and this position, this base attached to sugar at carbon number 1. Then this base and sugar, this is called ribose sugar. This is your, take it ribose, means your OH, HOH. This is called nucleoside, base and sugar. But nucleotide mainly, nucleotide structure, if you see the structure of nucleotide, mainly here, your sugar with base, then phosphate, then you can see the sugar, then base, like this. So this is one called nucleotide. This is called nucleotide. If it will be n number, that is called polynucleotide and forming nucleic acid. So, and there is one question: phosphate attached to sugar in which position? Here, sugar, phosphate, both sugar and phosphate linkage, that is nucleoside. That is sugar and base, that is your nucleoside. base, sugar and phosphate, that is nucleotide. Here sugar and phosphate forming the sugar phosphate. This bond here, phosphate attached to sugar in carbon number. Here is carbon number, you can say that is your C3 and C5. C3 C5 position, and base attached to sugar in C1. Here this position attached to base. Here base is attached to C1 position and C3 position and C5 position. Here this position phosphate linkage. One end is phosphate, another end is phosphate. So here if you see the phosphate here, this is also phosphate. This is your C5, this is your suppose C3. C3 dash, C5 dash, C5 dash, C3 dash, like this it is attached. And base attached to sugar in carbon number C1. Carbon number C1. And this bond is called, base sugar bond is called N-glycosidic linkage. N-glycosidic bond. N-glycosidic linkage. And here phosphate attached to sugar. In both end attached sugar, that is called phosphodiester. Phosphodiester bond. Phosphodiester bond, and it is called N-glycosidic linkage at C1 carbon, but here C3 and C5 dash. C3 dash and C5 dash. One side is C3 dash, another side is C5 dash. Like this, phosphate group is attached to sugar. So this is the structure of nucleic acid. Then the nucleic acid, the chain of nucleic acid attached to another chain. If you see, suppose this is the nucleic acid chain. This is the nucleic acid chain. Here what happens? A with the adenine with guanine. Adenine with thymine. Adenine with thymine. AT forming 2 hydrogen bonding, and cytosine with the guanine. CG forming 3 hydrogen bonding and AT forming 2 hydrogen bonding. So the chain, nucleic acid chain, linked one another. It may be separated in different form during the replication of DNA, formation of during division, cell division. But A attached to T and C attached to G, always in the form of hydrogen bonding. This is the hydrogen bond. This is also hydrogen bond. This is the hydrogen bond of between sugar C, G and AT in nucleic acid. So this is about the structure of nucleic acids. Then we will discuss the very important thing is biological significance. In biological significance, first important thing is that replication. During cell division, that formation of duplicate copy of DNA, duplicate copy of DNA, that is called replication. DNA divides into 2 similar copy. Duplicate copy formation, separation of like this, separation of DNA. One strand, another strand will be separated from one another. That is called replication. Separation of DNA strands into single strand, and the same copy of DNA will form. That is called duplicate copy of DNA formation is called replication. Then another important thing is, so biological significance we will discuss. First one is replication, that during formation, that the DNA divides into 2 equal halves. The DNA strand, the coil shape of DNA will be divided into 2 duplicate copy. That is called replication. Formation of duplicate copy of DNA is called replication. DNA, the DNA can be converted to RNA. During protein synthesis, first DNA convert to RNA. That is called transcription. This process is called transcription. Conversion of DNA to RNA is called transcription. And RNA, last point, the RNA synthesized protein. This process is called translation. Very important. Translation means formation of protein from RNA, that is translation, and RNA from DNA, that is transcription, and separation of DNA in duplicate copy, that is called replication. And all together, that is the hereditary character transfer from cell division like this takes place, but transfer of hereditary character from one generation to another generation due to the nucleic acid. When the nucleic acid play the role, because the chromosome mainly contains protein and nucleic acid. So the chromosome contains nucleic acid and protein. Two parts is combination of nucleic acid and protein. So nucleic acid mainly responsible for hereditary character, one generation to another. So this is about the nucleic acid, DNA, RNA. The main difference of DNA and RNA is here the DNA contain deoxyribose, RNA contain ribose sugar, and DNA contain AGTC bases, nitrogen bases, and RNA contain AGUC, only in place of T, U is present in RNA. And this is the main important difference. And the DNA is a double strand, it is a single strand, RNA is single strand. DNA is a double strand, two strands connect to one another, join one another, and the RNA is a single strand formation. And mainly protein synthesized by the RNA. RNA is protein synthesis, and DNA mainly responsible for transfer of hereditary character. So this is about the nucleic acid structure and biological significance and difference between DNA and RNA. Okay, thank you all.